Patient. Jens has published around 1,900 papers and is one of the physiologists with the highest H factor. And Jens has worked with gastrointestinal hormones for longer than I have been living. Actually, Jens, he wanted to become a surgeon, and I'm sure this would have benefited a lot of people with gastrointestinal diseases. But rather, Jens set out to help millions of patients around the world by demonstrating that a peptide from our small intestine that is released every time we eat actually have insulinotropic effect. Subsequently, Jens, he showed that super physiological levels of GLP-1 can mediate glucose lowering effects in patients with type 2 diabetes. And he finally also showed that this peptide has appetite regulatory mechanism. Today, these drugs are actually used worldwide for the treatment of obesity and type 2 diabetes. Perhaps what you do not know is that he also contributed to identifying the peptidase responsible for GLP-1 degradation, DPP-4. And together with several other scientists showed that DPP-4 inhibitors can actually also act as a glucose-lowering therapy. And that is actually a first-line treatment for type 2 diabetes. What I find personally with Jens, both in science and as a mentor, is that he tests his hypothesis in physiological experimental models, mostly perfusion models, and then he tried to translate potential findings into humans. So I would say he's a true translational scientist, but has also contributed to the education in science by training more than 100 students, including myself. This year, Jens, he will be awarded the Banting Medal for Scientific Achievement at the ADA. And what is the Banting Medal? It was actually given, or it was in the memory of Sir Frederick Banting, a key discoverer of insulin. And we all know that this is actually the year for discovery of the 100 years discovery for insulin. If you go into Wikipedia, and I've just sent you the Zoom link here, you should acknowledge, you should see who else have received the Banting Award. And we have a lot of people. We have one of my favorite, that is Elliot Joslin, who was one of the founder of Joslin Diabetes Center in Boston. And there are a number of known faces here. And now Jens also becomes one of them. So with these short words, I hope you will enjoy it, Jens. I will for sure. So let's have some fun.
Okay, so thank you for this very kind introduction. Yes, it is true, all that you said. So I'm going to give a presentation here, and you actually, or somebody gave me the title, How to Discover New Diabetes Medicine, and that's of course quite a challenge. But let me tell you 3 short stories about this. So let's have a look at this. This says how it all started, and these are cells in the gut mucosa and the gut epithelium that are stained with antisera against glucagon. And that, of course, is not as it is supposed to be. There's not supposed to be glucagon in the gut, and it's supposed to be in the pancreas, of course. So this was kind of, a provocation. It was challenging. What are they doing there? And many years ago, when I started, there was my first mentor really is Lisa Heating from the Novo Nordisk company. At that time, it was Novo. And Lisa Heating was working with glucagon, and she was very interested. Glucagon was also produced by Novo Nordisk, and so they were interested. Now I'm going to reduce this so I can see what I'm doing. Okay, and what she found was that she raised antibodies against glucagon, and she found that there were different types. And one of them, as you can see in this, reacted with extracts of the gut mucosa. That's what this says here. And the other one reacted with pancreas and did not react with extracts of the gut mucosa at all. So there were clearly 2 different types of antisera. And what was more was that this gut glucagon thing had to be different from the pancreas glucagon thing somehow. So that was her major discovery. And I was fortunate enough to be coupled to this very quickly to this development. What happened was that Lisa Heating, that the Novo company ran into a problem with the washing powder enzymes, And she was then denied to work further on with these glucagon, very interesting glucagon findings, and she was put onto washing powder enzymes instead. So that terminated her career. But before that, she just managed to publish this interesting study, which is a study of glucose administration to people. And you can see that this gut thing that you could measure actually increased when you took glucose by the mouth, whereas the real pancreas glucagon, it fell. So there was something, these cross-reacting, they could react with something from the gut that was different from what happened in the pancreas. So this was really the beginning of all this. And we started in a small group here with Jens Rehfeldt. He was at that time just a PhD student, but he had a knowledge of radioimmunoassays, and we came together and looked at patients with reactive hypoglycemia. And you can see the reactive hypoglycemia here. I was working in a department of surgery, and we had these patients, and it was quite a problem for them. And we also found that this gut glucagon thing, it increased in parallel with the insulin secretion. So we had this idea that the increased gut glucagon was a pathogenetic factor in the postprandial reactive hypoglycemia. And I must say that Jens Rehfeldt was my second important mentor here, and introduced me to the gut hormones and to the radioimmunoassay, which was a new and exciting technique. So some years passed by, and together with a number of people, particularly Alistair Moody and Lars Tim at the Novo company, we managed to find out what was in those cells that were lighting up with the antibodies against glucagon. And it was this molecule, which is called glycyntin, 69 amino acids here. And in the middle, it contains the in full glucagon sequence. And that was, of course, the explanation for the reaction with the glucagon antibodies. So it was truly glycyntin that was present there. And Alistair Moody and we proposed and showed in a quite convincing manner that this had to be at least part of proglucagon, because exactly the same pieces and bits here were found in the pancreas. One of these molecules down here was interesting, and that's the molecule that contains the glucagon sequence and also this little tail. That is what we called peak 2 glucagon, but of course it's a much better name to call it oxyntomodulin, as my friend Dominique Bataille called it. And that was an interesting peptide, and you can see that here because it did actually stimulate insulin secretion from my perfused pancreas preparation, together with Furio Baldissera back in the days. And there was quite a nice stimulation of insulin secretion. There was a problem with glucagon, as you can see, it was increased. That was not the great idea here. And we could show that the concentrations of oxyntomodulin were very, very low, so it was hardly those that were responsible for the reactive hypoglycemia. But the real breakthrough came when Graham Bell cloned the pre-proglucagon gene from hamsters first and from people afterwards. And the sequence looked like this. Sure enough, it was true with the glycyntin part that we had discovered already, but there was another part, and it contained not only one, but actually two GLP-1 and GLP-2, that is glucagon-like sequences, GLP-1 and GLP-2. And of course, we had them synthesized immediately and put them on our perfused pancreas, and they did not have any biological activity that we could measure, at least. So that was, of course, an extreme disappointment. So here's the pancreas that we were using in those days. This pot was my collaborator Sten Lindkjær's mother's potato pot that we were using for these experiments. Generally, we were helping ourselves with adhesive tape and chewing gum when we were doing our experiments in these days. But this pancreas is a very nice preparation, and it produces what it's supposed to. It produces pancreatic juice, but it also produces insulin. And then with our newly developed assays, we pulled out GLP-1 peptides from the gut extracts. And this is what was shown here. And then we looked at insulin secretion, and you can see that one of these peptides was a very potent insulin releaser. And that, of course, was extremely interesting and exciting. So eventually, through the work of Katrine Ørsgaard, who worked a lot on all this and did her medical thesis on it, we found out what the exact structures of all the components of proglucagon that we could find in the pancreas and in the gut, and actually also in the brain. And we found out exactly what GLP-1 was. It was a peptide corresponding to the sequence 728 to 107. It was amidated, and that was the real peptide. And this was the one that was also stimulating insulin secretion. We also found out what the real GLP-2 looked like, and that's another and very interesting story, but we can take that on another occasion. But so we managed to map this in full detail. So eventually we were working on all the functions of this new peptide, and that was, of course, extremely interesting. It had all these marvelous effects on insulin secretion and also insulin biosynthesis. So it didn't only stimulate secretion, but also stimulated the production of insulin from the pancreas. And it upregulated all the genes that are essential for beta cell function. There was a lot of fuss about the growth effects, and it could be demonstrated that it also would induce beta cell proliferation. Now, that discussion stopped a few years later when there was an accusation that the GLP-1s might cause cancer. So people very quickly forgot all about beta cell proliferation and all that stuff. But there was a little bit left of beta cell apoptosis, and that was actually very nicely demonstrated. So there's still a possibility that the GLP-1s act on beta cell apoptosis. And then there was this business of glucagon secretion, which was inhibited. We could show that in our pancreas very early on. And that is, of course, also interesting. And then eventually followed gastric emptying and satiety and appetite and food intake and weight loss and all that. And there were also beneficial cardiovascular effects, and I'm not going to talk more about that. There were improvements in insulin sensitivity, and Mette Sandler, that you see here, kind of collected the whole thing in her Lancet paper, where we actually managed to look at patients with type 2 diabetes and infuse them with GLP-1 in an infusion pump for 6 weeks, and could show all of these very nice effects, because you have all the problems of the type 2 diabetics over here, and it actually worked on all of these parameters. So this was really a major effort, and it was fantastic. And Mette Sandler, all her patients, she celebrated her findings here with her PhD thesis and a party, and her patients came and did the cooking because they liked her so much. We all did, by the way. So how did that story end? Well, it ended up with some of the newer drugs, and I'm just showing you this fantastic figure of the effects of tirzepatide, which is the GLP-1 GIP co-agonist developed by Lilly. And what you see here is really sensational, because what you see is, in this group with this dosage here, patients with type 2 diabetes, 50% of those that were treated managed to get an A1C level below 5.7%, which is normal. It completely outperformed semaglutide at 1 milligram, but of course they didn't include the 2.4 milligram dose, which is used today, and that must happen at some stage. But so far, what is important is to note that with these therapies, you can now have more than 50% of the patients at completely normal A1C levels. And at the same time, the weight changes are really impressive, again, outperforming the effects of semaglutide. So that's how far this story goes currently, and it's, of course, extremely encouraging. Now let's move on a little bit. So during these stages of development of GLP-1, we injected, of course, patients with type 2 diabetes with GLP-1, and we looked at how well it survived in the body. And as you can see here, very, very, very little of GLP-1 survives in the intact form, whereas almost all of it is metabolized very quickly, so that it's really a waste of good peptide to inject this subcutaneously in people. This was the maximum tolerated dose that we could inject, and sure enough, it didn't work very well at all. And that's where Carolyn Deacon comes into the picture, because she and I decided to look at this metabolism of GLP-1. And she did a lot of studies with various inhibitors and mechanisms and so forth, and a lot of chromatography and what have you, HPLC. And we found out that there was one enzyme that was clearly responsible for this, the enzyme dipeptidyl peptidase 4. And it cleaves off these two amino acids here. And this part of the molecule, the metabolite, is then rendered completely inactive. And this happens with an incredible rapidity of so that the half-life is just 1 or 2 minutes in people. This was a very dramatic effect, of course, but it also led to very quickly to this idea that inhibition of this enzyme could prove a useful adjunct in the management of type 2 diabetes. As was the case, we were completely inspired by ACE inhibitors to treat hypertension, of course. So this was the idea that we put forward then back in 1995. And sure enough, we started doing experiments in pigs to have a look at this. So we had some anesthetized pigs here, and we infused them with GLP-1. And then we also gave them a little bit of glucose. And then we could give them valine pyrrolidine, an inhibitor of the enzyme. And then we would repeat the GLP-1 infusion and the glucose infusion and see what happened. And what we're doing here is that we are measuring GLP-1. So this is the intact active GLP-1 during the infusion. And you can see that it's broken down tremendously in spite of continuously infusing GLP-1. It's still broken down very extensively. That's how serious it is. But with this inhibitor here, we could actually completely prevent the breakdown of GLP-1. Now the two curves are completely superimposable. All of it was protected. And we will just note this valine pyrrolidine and remember this question: where did that come from? How come we had such an inhibitor? Okay, but what then happened was that we looked at insulin secretion in these animals, and you can see as soon as we gave the inhibitor, well, then we have a protection of GLP-1, the concentration, that's the endogenous one, increases. And here we have the infusion, and we have a tremendous increase in the secretion of insulin, a 3 to 4-fold increase. So this really worked, this idea of inhibiting the enzyme. Not only could we protect the peptide completely from being degraded, but also we could increase the insulin secretion by 3-4-fold. So this was really something. And the idea was incredibly rapidly taken up by the pharmaceutical industry, and very quickly they were able to synthesize various inhibitors that could be used for patients, of course. Here is vildagliptin and sitagliptin and saxagliptin, and they were able to inhibit the enzyme for about 24 hours of the day, and that caused, of course, a very important protection of not only, it turned out, GLP-1, but also GIP. So that's so where did the inhibitor valine pyrrolidine come from? That's an interesting story. And so the truth is that one of the first companies to engage in this was Novartis. And Novartis, they talked to us and we discussed it with them, and we did the first studies together. And why is it that they dare to use inhibitors for diabetes? Because in those days, if you had asked an immunologist, what about treating diabetes with an inhibitor of DPP-4, he would say to you, you must be out of your mind. You are completely crazy. And that's because DPP-4 was known, it's also called CD26. And it was thought to be involved in antigen presentation in the immune system. But the secret was that Novartis in Basel had, there's a double echo now, had been developing inhibitors for DPP-4 because they were interested in anti-immune therapy. So in Basel, they had a lot of these inhibitors. And it turned out that the more specific and selective they made the inhibitors for DPP-4, the poorer they inhibited the immune reactions. And finally, they had some inhibitors that were completely inactive in the immune system, but were still very active in inhibiting DPP-4. So they knew that this could be done. And that's why they, as the first company, dared to do this. And all the other companies, they were just spying on them. And this is true for Merck, certainly, which then eventually produced the first, the first very good inhibitor, sitagliptin. So what happened then, and you all know that, of course, is that they have been tremendously successful in diabetes therapy. And actually, today, the DPP-4 inhibitors have the largest market share and a tremendous revenue in sales. So it has been a tremendous success. Sitagliptin will run out of patent in next year. And that will mean, of course, that the price of the DPP-4 inhibitors will go down precipitously. And that would be wonderful because then many more patients can afford to use these very useful medications. Now, on to the third story. So here we are looking at what happens after one wide gastric bypass, which is something that we've been very interested in. And why is that? That is because after the surgical reconstruction here, food rushes down the small intestine and reaches part of the gut where there's a lot of GLP-1 and also the peptide YY. And that causes a very exaggerated secretion of these two peptides, and that then has all of these effects on insulin secretion and on food intake. So this we looked at in great detail, and Niels Brun Jørgensen from Hvidovre working with Sten Madspad also. Madspad has been behind almost everything we have done. He looked at these patients, both with type 2 diabetes and with normal glucose tolerance, before and after the operation, and we found these tremendous increases in GLP-1 secretion after here 1 week, 3 months, and 1 year after the operation. Tremendous increases. And this, of course, was extremely exciting. So what we did was then to treat patients with Exendin 9, which is a GLP-1 receptor antagonist, and look at what happened. So this is what happened with glucose tolerance here after the operation. So this was before the operation, and then you can see the improvement after 1 week and the improvement after 3 months. And it's truly a wonderful effect on diabetes. As you can see here, diabetes, and then it's much better. And now the diabetes is really gone after 3 months. But then if you block the actions of GLP-1, what happens? You impair glucose tolerance before the operation, but certainly also after the operation. And if you express this as AUC for the glucose under the curve here, you can see that all that was gained by the operation is lost when you add Exendin to the profile here. All that was gained was lost, and you can see that now the areas are completely the same. And that, of course, was because of the effect on insulin secretion, here shown as insulin secretion rates, the insulin secretion before, and then you can see the increase after, and then you can see the large increase in insulin secretion and the left shift in the profile, and you can see how this is impaired if you block the effects of GLP-1. This is what you see here. And then you can see if you take the AUC here, that all that was gained by the operation here and here is lost after blocking the GLP-1 receptor. So this is truly an effect of GLP-1 secretion. Then looking at food intake, what happens here? And again, we have the entry of nutrients here coming down to the small intestine, and this huge release of GLP-1 and PYY. So we were wondering, what is the importance of GLP-1 and PYY for this food intake reduction that happens, and the reduction in overweight and everything. And Maria Svane, also working at Hvidovre, did these very nice studies. First of all, we ensured that GLP-1 and PYY would really have an effect on energy intake. And we found this interesting thing that with low doses of both hormone, these are infusions, there would be not only an additive effect, but apparently a synergistic effect on food intake here. So they are truly inhibitors of food intake, particularly when given together. So what she did was this interesting experiment. She could block the effects of GLP-1, of course, with Exendin 9. That was this way. But for PYY, we didn't have an antagonist. Now it turns out that PYY is very interesting. It is secreted as an active peptide in the form of PYY 1 to 36. But then it must be broken down to PYY 3 to 36, and that is active on food intake. We didn't have any antagonist, but this breaking down here, this metabolism is catalyzed by dipeptidyl peptidase 4, the one we have just talked about. And for those, we have, of course, sitagliptin. So we could block the DPP4, and then we could prevent the formation of PYY 3 to 36. And we could show in our studies that this is exactly what happened. So here, block. And what happened then to food intake? So placebo, this is what the food intake was. And these are operated people, of course. And then we gave them Exendin 9. Did that work? No, it didn't. And then we gave them DPP4 inhibitors on their own. That didn't also work. But together, we had a 20% significant intake increase as expected, showing that the two hormones were responsible for keeping down food intake by 20%. And that's exactly the figure we needed to explain the reduction in food intake and the reduction in body weight. So that was that story. So now comes the question, why do we have, so again, a question that must be answered. Why is it that we have this tremendous rise in GLP-1 secretion? How is it actually regulated in the gut? Who says that the gut should all of a sudden produce a lot of GLP-1? And here we have been very interested in a particular mechanism here, a paracrine regulation of the L cell. The L cell is the cell that produces the GLP-1 here. And here, Sarah in our laboratory has been doing all these studies for her PhD thesis, and also she's working on it now, as I'll show you. So we have the L cell here, and it produces GLP-1. And the idea is then that GLP-1 diffuses to neighboring D cells. They produce somatostatin, and they are equipped, we have shown that, with a GLP-1 receptor, and that can be activated. And when that happens, then they will produce a lot of somatostatin that will then feed back onto somatostatin receptors. And we've found out that the L cells in particular express the SSTR5 subtype. So what we have is this link between the L cell and the D cell. Once this is stimulated, it will stimulate secretion of somatostatin, which will then inhibit L cell secretion via the somatostatin 5 receptor antagonist. So if you block this somatostatin 5 receptor, then you should be able to increase GLP-1 secretion. So that's the point. So what she's been doing is to work with the isolated perfused mouse intestine, and you see it here. It's very small. You can barely see it. It's terrible, but she's very good at it, so it works anyway. And then she can infuse it with somatostatin 5 receptor antagonists, and we can see what happens. So here, first of all, we show that somatostatin, it exists in two forms. Particularly this 28 form is produced in the gut, and both 14, but particularly somatostatin 28 is very powerful in inhibiting GLP-1 secretion from the gut. And down here, we are adding the somatostatin 5 receptor antagonist. And now you can see that for dose response here, that you can increase the secretion of GLP-1. So as you increase the secretion of GLP-1, of course, you will also increase, as I told you, the secretion of somatostatin. But because we have blocked the receptor, there is no inhibition. There is an increase in the secretion of GLP-1. So there's a tonic inhibition of GLP-1 secretion going on, and this is very important, of course. And also you can completely break this tonic feedback regulation with a somatostatin 5 receptor antagonist. And here is the key result really of the studies. So here we are feeding the gut with luminal glucose. That's the stimulus for GLP-1 secretion, of course. And this is the normal, very nice stimulation of GLP-1 secretion from the perfused gut. But then if you add a somatostatin 5 receptor antagonist on top, then you can see that you can magnify this response very, very much. So this is, of course, interesting. So what she did then was to look at a number of mice here. And here she's treating the mice with either GLP-1 or with a somatostatin 5 receptor antagonist. And you can see that you can get a powerful improvement in the oral glucose tolerance. And here, very interestingly, here we are looking at the effects of DPP-4. Why add DPP-4? Because what DPP-4 inhibitors do is, of course, to protect this newly released peptide. So much more of it survives in the circulation. And here you can see the control experiment. And then after it has been given the somatostatin 5 receptor antagonist, I'm sorry. And then you can see the effects of the DPP-4 inhibitor itself. It also helps. And then the combination, which causes a very pronounced improvement in glucose tolerance in these animals. And here we are trying to show that it is truly an effect of GLP-1. And what do we have here? We can use exendin 9 to block the effect here. And at the same time, these are somatostatin 5 receptor knockout mice. And over here, you can see what happens in the small intestine. You can see that it produces an enormous amount of GLP-1 because it doesn't have the somatostatin 5 receptor to cause the feedback. And here you have that they have better glucose tolerance here. And then if you take away the mechanism, the GLP-1 operated mechanism, you lose that glucose tolerance completely. So all of this proves that it is GLP-1 really that does it, and that it acts via the somatostatin 5 receptor. And finally, we have looked at diet-induced obese mice, of course, and here you can see control mice, what happens, and then you can see that you can also, with these somatostatin 5 receptor antagonists, improve glucose tolerance. So the whole important thing here is that we are now going on with planning clinical studies with a somatostatin 5 receptor antagonist for oral use, and we will use it together with DPP-4 inhibitors, and we expect that we can use them for diabetes and hopefully also for treatment of overweight. And they will be tablets orally available, and we profoundly believe that we will not have any problems with side effects. So this is going to be very, very interesting. So these were the three stories that I wanted to tell, how to discover new diabetes drugs. And what is the most important? Well, I think perhaps judging back from all the work we've been doing, the most important is really to collaborate, to establish good collaborations. Don't be afraid to collaborate. Find good collaborators, make friends, don't be crazy, work together instead. And then perhaps it's a good idea to remember your physiology, because if you don't remember the physiology, how can you get good ideas? And then, very important, be curious, be curious. And then ask questions to all of the things you observed in the laboratory and in the clinical work. Why is that? Why on earth does this and that move in that direction? What is it that happens? So ask questions. Don't listen to professional advisors. They're always wrong and very boring to listen to. So don't do that. And first of all, don't give up. So thank you. This is what I wanted to say today.
Okay, so thank you very much, Jens. That was a lot of fun and great talk. So I just wanted to ask the audience for questions. I can see there's clapping hands, so that's nice. Perhaps I can just start myself. So you started with the story on GLP-1, and now there's also some support that perhaps these agonists of the GLP-1 receptor could be used for treatment of Alzheimer's, and what do you have? So where do you think that this compound or these compounds will take us? Will that also include dementia and other diseases, or what's your opinion?
So first of all, I'd like to say that, from the day this idea about the GLP-1s began to catch on a little bit, the number of proposals as to what it does has been incredible. There is not a mechanism in the body that has not been reported to be influenced by GLP-1. So it's been a little bit of a hard work and a little bit of a nightmare to clean up all these reports from out there and to try to see if all of that is correct. But one of the problems with the GLP-1 receptor agonists is to get them to the brain. And for that, we have this blood-brain barrier. We know rather precisely where some of the agonists that are used today, how they enter the brain via the leaks in the blood-brain barrier. And we know rather well where they enter and what nuclei they actually affect on. So if you want them to have a protective effect on neurons in the brain, you really have to get the compound up there. And that is a major problem, I would say. But on the other hand, I must say that the improvement in metabolism with the newer GLP-1 receptor agonist is quite profound. Their effect on inflammation, whether it's direct or secondary, their effect on atherosclerosis, their effect on all sorts of metabolic parameters, they're really impressive. And I find it completely natural that once you improve circulation and metabolism and inflammation, that you will also have beneficial effects on something like Alzheimer's disease. I would love to see a specific effect of the GLP-1 receptors on Alzheimer, and there are studies going on right now, and I'm quite positive that they will come out positively. But the direct mechanism, I just, I'm open. I love it, but I can't see right now how it's going to happen.
Okay. So I haven't noticed any questions from the audience. You are more than welcome to speak up or or raise a hand or write in the chat, please. So meanwhile, I will just give it another go here. So
You're
Jens,
welcome.
you've been in this game for, I don't know, 40
No, no.
years, 50 years, I don't know, many years at least. So this is also meant, this symposium, to kind of inform the younger scientists as myself. So what's your opinion? Have science and how we conduct science, have that changed for the last 30 years, you think?
Yeah, and you can contrast Juline's wonderful lecture to mine. Hers is very much based on omics and how to handle modern collection of big data, whereas all I've shown you here is really hunting, chasing a single hypothesis. And there's one difference that I really must emphasize. The omics approach compared to my approach, the hypothesis-driven, is much more expensive. Really much more expensive. And that doesn't mean, of course, that you shouldn't do it. It's just that, and also the payoff, the results. If every time, if you're going out to fish for a very special fish, and to get this very special fish, you have to fish up all the fish in the ocean. It's going to be a major undertaking, right? And maybe with a bit of luck, you find your single fish, but you can't be sure. So whether it's cost effective is really a problem. And then I wonder if you're just one of the little wheels in this huge machine collecting data, if it is as fun as it has been to chase a hypothesis. To chase a hypothesis, that's where you sometimes really feel that you do some thinking, that you try, like Sherlock Holmes, to put two and two together and see if you can come up with a good idea as to how to proceed. And so I think this has been, almost half the fun, or perhaps even all the fun. And if you take that away, then I'm not so sure it's so fun any longer. But of course, if you get a shining result and you're author number 263 on the Nature paper, that has also some pleasure, of course.
Okay, so we have a question from Nils Fairman. He right here, we increasingly see that there are many small metabolites that also regulate insulin secretion. How much do you think these smaller metabolites contribute to the overall effect on insulin secretion compared to GLP-1?
I'd answer it this way. So one of the most impressive studies to my and results here during the last few years has been the effect of weight loss in diabetes. I'm particularly thinking of something like the DIRECT study, where Roy Taylor managed to make some Scottish people eat less, and I guess they could save money in that way, and that's perhaps why this was successful in Scotland and nowhere else. But nevertheless, they managed to do it, and that diabetes disappeared. It was really amazing. If you could produce a weight loss of 15%, and they did that, some of them did, quite a number of them did, then 85% of them would have a remission of diabetes in that group. That is amazing. And the same is really seen after bariatric surgery. So my absolute key focus here is that it is not so much, of course, type 2 diabetes has this genetic component on beta cell function, which is a key problem. But the other problem is the overeating. And the overeating may very well have some genetic components and psychological components and all sorts of important things. And that is, of course, what we really want to know something about. But the fact is that if you stop eating, overeating, then you will have a remission rate which is tremendous. And that, to me, that tells me that you don't really need to have all these intricate little mechanisms. All you have to do is to make sure that people don't eat as much. That is my key light ahead. That's my beacon in front of me. Yeah, that's what I would like to answer on that.
Okay, thank you. So there is a question here from Frida. So thank you for a wonderful talk. The anti-inflammatory effects of GLP-1 you mentioned, could you elaborate a bit on that? Is it a direct effect of GLP-1 or through weight loss?
Again, as I said, people have been incredibly inventive and imaginative ever since it started catching on in finding mechanisms for GLP-1. And they, as you know, receptors have been identified almost everywhere in the body. So it has been, again, a little bit of a nightmare to try to clean that up and find out where the receptor is actually expressed. So one of the sites where we constantly find it today is in the endothelial cells. And that, of course, is interesting. So maybe there's something there. And there's also a link to the immune system. We have these intraepithelial lymphocytes in the gut that seem to link something with the immune system, GLP-1 secretion with the immune system. And that, of course, is another very interesting lead. But otherwise, that I think that what convinces me that there is something is that in the new studies with semaglutide, 2.4 milligram, there are quite pronounced reductions in CRP. Really quite impressive, even if they're low already, but you know, high sensitivity CRP, you get really quite pronounced reductions. And I just wonder whether this is a key to a better understanding of what goes on. But apart from that, there's no doubt that if you take away an obesity burden from people, and you know that is supposed to be accompanied by inflammation in the adipose tissue, if you take that away, well, then perhaps some of the inflammation goes away also. And again, the overweight and obesity is a key issue here.
Thank you for your answer. So there is a final question here from Luis Rodigo. Hope I pronounced it right. Thank you for such an inspiring talk. It has been controversial about if the molecular mechanism of GLP-1 to amplify insulin secretion also involved better performance of mitochondrial metabolism in beta cells. Do you think there is a real effect of GLP-1 on beta cell mitochondrial metabolism to amplify so glucose stimulated insulin secretion?
That's a good question, and I cannot answer it. I don't think so. You know, I have friends in the tower and elsewhere in the Panama Institute, and the people have been carrying mitochondrial respiration apparatuses across the inland ice in Greenland several times, and you know the most incredible efforts have been done to look at mitochondrial problems in type 2 diabetes. And you know, my take on all these many studies is that this is not where the key issue is. It is not where the key issue is. The key issue in type 2 diabetes is the genetic problems in the beta cell. And whether they hit mitochondria, that is possible, but I don't know. And the other key issue is, again, that if a person with a genetic problem in the beta cells develops obesity, that's when we have the problem. That's when the type 2 diabetes develops, and when we have the hypoglycemia and the hypoglycemia and all the problems that arrive. So again, food intake and whether, you know, is a key.
Okay, so there is a comment or a question, I'm not sure, by AU478460. No, so perhaps you could either speak up because you wrote...
That's a Chinese spy, I'm sure.
Okay. The proliferation of beta cell by GLP-1, can that be a medication to continue working on? I think that's how I interpret the comment. Yeah.
So the data I usually use to discuss this issue is the LEADER study, which is the cardiovascular outcome trial with liraglutide. But that's because it was a very long study, up to 5 years of observation, and where you know, people were followed with A1C measurements over all these up to 5 years. And the curve shows that there is almost a completely maintained improvement in A1C levels over all of these 5 years. There was also a good result in the placebo group, but in the placebo group, this was a blinded study. People were taking a lot more insulin and other anti-diabetic agents. So it is truly a very remarkable effect. And you wouldn't think, if you look back to ADOPT studies and other long-term progression studies, you wouldn't think that people with diabetes would be able to maintain good improved glucose tolerance and A1C levels for such a long period without deterioration. So the only way to interpret that actually is to say, well, there is somehow a protective effect. There's never been a sign of proliferation in people. There was a very nice study with exendin 4, with exenatide, where people looked directly at beta cell function and did a beta cell challenge test over a 3-year period, and there was no improvement. There was no sign of an increased beta cell function over these 3 years, but it was maintained again. And you wouldn't think that people with this disease were able to maintain a beta cell function for such a long time without something going on. So I'm in favor of a protective anti-apoptotic mechanism, which has been shown very nicely. Whereas with the grown-up beta cells, I think this is consistent with animal data as well. They're not responding with proliferation. You have to have very, very young beta cells to show this.
Okay, I cannot see any questions or further question. I just have a final one. So you mentioned the GLP-1 GIP co-agonist, right? With the beautiful data on A1C. So my question will be that now we see a market that is getting filled with all sort of co-agonists, triple agonists, unimolecular triple agonists, and so forth. So do you think these compounds are just super GLP-1 agonists, or are there actually some effects from GIP agonist or perhaps antagonist? What do I know?
Yeah, so first of all, so tirzepatide, which is very interesting and probably going to hit the market very, very soon with great success, I must say, is a GLP-1 GIP co-agonist. And there are other GLP-1 GIP co-agonists. Novo Nordisk actually developed one, and they had it in clinical trials. And they even published it in, I think it was in Molecular Metabolism, something nice. But it was very bad. It was very bad. So, because you combine GIP and GLP-1, you don't necessarily get a very good drug. So something special has to happen. We have been able to work with tirzepatide, and it's very interesting because we can show that the interaction of tirzepatide with the GIP receptor is completely identical to that of natural GIP with the GIP receptor. But the interaction of tirzepatide with the GLP-1 receptor, on the other hand, is very, very abnormal. It does not induce arrest in recrutation, and it does not cause internalization. So the interpretation is that perhaps it keeps the GLP-1 receptor on the surface of the cells and therefore activates the receptor even better. So my current interpretation is that it could very well be a super GLP-1 agonist, tirzepatide. But the people at Lilly, and we did the work together with those people, they don't agree. They interpret the data completely different. And I guess this is because their whole story would fall to the ground if it is true. But those were the findings.
Okay, thank you very much.